Okay, I think we can start. It's already like 4 minutes up with three. I think around 10 of you, around 9 of you joined. Thank you for joining. So like I don't know whether if I think around 35 are registered for exam for this course. So if not, I think maybe some of you might join as well. So instead of just waiting, maybe I can just go through what kind of background you people are having, so that accordingly how deep or how normally I need to go to the course, go with the course, I can accordingly analyze that. So can you all introduce yourself? Like you can unmute yourself or just write it in the chat box. Like introducing in the sense where, what is your background basically and what are you currently studying? Yes, if you want, you can unmute or else just write it in the chat box.
Yeah, hello Gaurav. Glad to meet you. Am I audible?
Yes, yes, you are.
Yeah, yeah. So I'm currently working. I'm just working as a researcher in Bangalore, and
Oh,
just the interest was protein work was my interest. I am a master's in biomedical genetics, and
oh,
protein interaction networks and all those was my interest. So just on the basis of interest, I have joined. Yeah.
oh, great, great, great. Nice to see you here. Yeah, anyone else who wants to introduce themselves? So he's like you are well versed with almost basics of it. So today's lecture will be mostly basics. It might be just repetitive for you, but for others, so there is Prityusha who is doing currently BSc Biotech. So maybe a little advanced, more basic will be for you for Prityusha. How about others? Oh, there are SRFs as well. There is someone from BSc and there is oh, you just finished your 12th grade, so like this lecture would be more important for you, Lekha, then. And there's someone from BTech as well, Sambhavi, and BSc Microbiology second year. Okay, so very, very diverse crowd, right from 12th grade, just finishing 12th grade, and all, and currently who is doing job as well. So like a very vast crowd. So I'll be dealing with both. So I need to go with basics as well as few of you are in currently doing BSc or BTech. So I'll be talking about basics as well. So maybe today's lecture I'll go a little in deep of basics, and as this is a 4 week course, maybe we'll be slowly going towards the advanced stages later. Yeah. So I think we can start with the lecture. It's almost 7 minutes up already. So I don't know if anyone else is joining. So whoever here are around 9 of you, we can start the lecture. Yeah. I hope my screen is visible as well. Yes.
Yes, yes.
Yeah. I'll just check whether I have started recording as well. Yeah. Okay. So this is one of the NPTEL course which has a code which is BT78, and it is called as Proteins and Gel-Based Proteomics. So this specific course is actually taken by Professor Sanjiva, and I am appointed as a TA for this specific course, myself, Gaurav Bingi, and I'm currently pursuing my PhD at IIT Bombay. So as we are, so this comes under PMRF fellowship basically. So as I am a PMRF fellow, we are supposed to do certain teaching. So NPTEL gives us an opportunity to do this teaching stuff. So in my PhD itself. So this is the course which I am allotted. So I am the TA for this specific course, which is proteins and gel-based proteomics. And so this specific lecture will be every Saturday 3 to 5 p.m. And so if any of your friends still wants to join the course, the last enrollment date is 28th July. So I think around 2 days are left. So if any one of you or your friends might wants to join this specific course and wants to get enrolled in this specific course, 2 days are left, so you can inform them accordingly, and then you can later, I think, register for exam as well, and the exam date is 21st September 2025. So this is just the details about the course, and to give an overview of the course, so today is 26th, so we'll be like week one lecture basically. I'm calling it as lecture one, or else week one we can say. And so this specific lecture, today's lecture, will be majorly on basics of amino acids and proteins. Uh, Basically, I'll try and give you overview of the whole course itself, uh, and a little bit more in detail about what exactly amino acids are and what exactly proteins are, and along with that, a brief overview of all of this a bit complicated topics like 2D gel electrophoresis or DIGE and mass spec, etc. Then this is what I'll be doing in this specific lecture. And in terms of week two, which will be on 2nd August, during that specific lecture, we'll be dealing with much more in detail about gel-based proteomics and sample preparation, how exactly samples are prepared for doing proteomics, and what all different methods are available for doing proteomics. But in that also we'll be majorly looking at gel based proteomics itself, and there are gel free proteomics as well, but we'll be dealing with only gel based proteomics. And in terms of that gel based itself, there is one of the ways is 2D gel electrophoresis. We can also call it as two-dimensional gel electrophoresis. And so in this lecture, you will be learning much more in detail about this specific technique. And in the week 4 lecture, we will be discussing more about DIGE, this difference gel electrophoresis short form is DIGE. And we'll be also talking about mass spectrometer, which spectrometry, which deals with the analysis of whatever protein might be present in your sample, and how can we determine certain biomarkers using those specific techniques. So coming to lecture one, so today we'll be majorly talking about all of this. So I've divided into five, basically five different parts, and even Professor Sanjeev himself has divided it into five different parts. You might, if you might have gone through the lectures earlier, I just wanted to know if you have all gone through those videos which are provided in the website. If not, you can also let me know about that. Have you all gone through those videos of week one lectures? Yes, yes or no? Even no is fine. Yes is also fine. Oh, Aditi and Pinky has gone through that. What about others? Okay, that's fine. Okay, a few of you have looked through the lectures. You have done it halfway, and few of you are just started. Okay, fine. So what I'll be doing basically in this lectures, like on 26th, 2nd, 9th, and 16th. So what I'll be basically doing in those lectures is, so I have gone through those videos already, and what I'll be doing is so those lectures are around 40 minutes to 1 hour or so. So what I'll be doing is within an hour or so, maybe with 1.5 hour, I'll try and cover all those lectures of week one, all those five lectures which has been taught to you in those videos. So I'll try to cover all those five lectures in around 1 to 1.5 hour, and remaining half an hour maybe we can discuss certain questions related to it. And if you have any kind of doubts with this week one lectures, we can discuss on that. So that will be the major idea of the whole 2 hours course, which is from 3 to 5. So that is what I'll be doing. Initial 1 to 1.5 hour will be just an overview of all those five videos, and later half an hour around will be discussing if you people have any kind of doubts, or and we'll be going through certain MCQs, certain questions, which will help you to solve your assignments. Getting it? All okay till now? Yes.
Yes.
Thanks. Okay, so let's start. So first thing will be introduction of amino acids. Then I'll then introducing proteins. Then how protein folding and misfolding can affect the protein function, and how this folding misfolding can cause different kinds of diseases. And if we have a protein inside a cell, how can we extract that specific protein? How can we purify that? We'll be dealing with that a bit, and a bit of introduction of proteomics. Yeah. So most of you all might be knowing it, but few of you who are doing BSc or just finished 12th grade, for them it might be uh, still you people might be knowing about it, but it will be a very good what can I say, like refreshment of the same topic which you have already done. And for those who have already working or already done BTech or MTech, for them it will be just a basic overview of the whole thing which you already know. So that will be helpful for you all, so that it brings up whatever you have already studied. Okay, so how generally an amino acid structure looks. So this is the structure of an amino acid, which has an amino group and a carboxyl group and hydrogen thing and a variable group. So this R is the reason by which we are having different amino acids. So and to the left there is amine group, there is carboxyl group, and there is R, which is a variable group. And along on the basis of that, there are different amino acids. There are around 20 amino acids present in our body, which is one of these are the examples of it: glycine, alanine, serine, etc. So if you can see here, uh, this is the side chain. You can see this is CH3, then CH3OH, CH3, CH3OH. So these are the different side chains. So the only R group is something which varies from amino acid to amino acid, whereas this specific structure is same for all the amino acids, the above one. So how can we classify these amino acids? So there are different ways of classification. One of the way is depending upon the polarity. If it is having certain charge, or if it is not having any charge, we can classify them into polar amino acids. And if they have certain sulfur containing aromatic, aliphatic, or basically which are non-polar in nature, those will be non-polar amino acids. So this is the one of the basic way of dividing them, but we can have n number of different ways to classify them, like acidic, basic, neutral. They are ultimately polar, but that's a different way of classifying. One of the different way of classifying is like essential or non-essential amino acids. Essential, so I'll be talking about all of those. So there are different ways of classifying amino acids, but one of the way is differentiating them as polar and non-polar. I'll just add the pointer as well, so that wherever I'm explaining, I can just go through that. So as we said, there are 20 different amino acid and there are different ways to classify them. So these are the classifications groups itself. So these are non-polar aliphatic groups. So this classification also depends upon the side chain which it has. So these side chains are non-polar in nature and they are aliphatic. So they come under aliphatic group of non-polar nature. And these are also non-polar, but these are having aromatic groups. So these like phenylalanine, tyrosine, and tryptophan, they come under aromatic non-polar amino acids, and these are aliphatic non-polar amino acids. And in terms of polar, there can be neutral, uncharged one, and there can be either positively charged or negatively charged. So positively charged are, for example, lysine, arginine, and histidine. So they have NH2 plus or NH3 plus, you can see here. So they are positively charged, whereas these two specific amino acid have a side chain with carboxyl group, which makes them negatively charged. So these are negatively charged amino acids, whereas these are uncharged amino acids, which are having branched kind of a side chain. And so these are examples of polar uncharged amino acid. These are positively charged amino acid, these are negatively charged, and these are aromatic non-polar. So this method of classification will give you this kind of classification. And so each amino acid is having are they have different types. There are different types of isomers for it. So they are divided into two, which is L isomer and D isomer. That specifically depends upon uh, the amino group which is there. So if amino group is on the left side, it is L isomer, and if it is on the right side, it is D isomer. So that's how we can basically detect it. The if or understand that if it is if amino group is in the left side, it's called as L isomer, and if it is on the right side, it is called as D isomer. But the main reason being them called as L or D isomer is because of the, I don't know if I'm having that image. Okay, I'm not having that image. So it's basically because of the um rotation of the polarized light. So if a specific amino acid is uh, uh, exposed to polarized light, what will happen? It will either rotate it like levorotatory or dextrorotatory, depending upon that. They are labeled as L isomer or D isomer. So it's the major reason being giving them L or D is because of the rotation of polarized light, how they can do it. Majorly, we can either find, so there are, we can either find L isomer as well as D isomer in our body. And generally amino acids, most of the amino acids are non superimposable, meaning so is the example itself is on the backside in your hands itself. So it can form mirror images exactly, but they are non superimposable to each other. And whereas there is only one amino acids which is achiral in nature, and they doesn't represent chirality. Can anybody answer that which is the amino acid which is achiral in nature and not and doesn't have any
Glycine.
chirality as such? No, not lysine. Anyone else wants to answer? It's not lysine though. Do you all want to see the structure of amino acid and then predict the answer? So this chirality depends upon the chiral carbon itself. When do we say a carbon is chiral? It depends upon all four groups are different. So the valency of carbon is 4, right? So it should have 4 bonds with carbon. So if all those groups are different, then that specific carbon is called as chiral carbon. So looking at these amino acid structures, can you all tell me which is that specific amino acid which is achiral in nature and doesn't have chirality as such?
Proline.
Not proline also. All four groups are different in proline too. So
Glycine.
yeah, glycine is the answer. So if you can see here, both hydrogen bonds, it's like two bonds are with hydrogen itself, so it doesn't represent, so it cannot form mirror image as such. And this is not a chiral carbon, this is achiral. So glycine is the only amino acid which is achiral in nature, whereas all other amino acids are having chirality. Yeah, so this is what I was talking about, polarity, like the optical property. And if they can form mirror images, they are also called as enantiomers. The L-alanine and D-alanine are enantiomers of each other. So if they can form mirror images, they can also be called as enantiomers, and the carbon is called as chiral center or a chiral carbon. So I was talking about this itself, the optical activity. So amino acids are able to rotate the polarized light, and if they are levorotatory, they are called as L-amino acid, and if they are rotating towards the right hand side, they are called as dextrorotatory, and they are called as D amino acids accordingly. So that's what I was talking about. So L and D basically comes because of the rotation of polarized light. Yeah, there are some other properties of amino acids. So there are few amino acids which can absorb certain wavelength of light, and majorly they are aromatic amino acids, tryptophan, tyrosine, and phenylalanine. So these are specific amino acids which are having maximum absorbance at specific wavelength. So that's the reason if you want to determine the concentration of any specific protein, or so we generally take that specific concentration at 280 nanometer, because That is, so these are in the range of 280 itself. So that's the reason we take protein concentration in 280 nanometer wavelength, because these specific amino acids observe wavelength at 280 range. So the one of the other way of classifying amino acid is they can be classified as essential and non-essential, indicating that essential indicating they cannot be synthesized in our body. That's the reason they are essential, and we need to take it from the food itself. Those are called as essential amino acids. And non-essential are the one which body can produce by itself. So we don't require them from food. Our body itself can produce those specific amino acids. So those are called as non-essential one, because we don't need them. Our body is capable of producing them, whereas essential are the one which we needed from our diet, and as our body cannot synthesize them. So this is other way of classifying amino acids. Similarly, there are a few other properties of amino acids. Depending upon the pH, the charge is different. So Pi, or isoelectric point, is a point at which the charge is neutral. So if you see the one in blue here, it is having positive and negative. So if there are two charges, positive and negative, they will cancel each other, and there will be no charge present for this specific amino acid at that specific pH value. So though that is called as isoelectric point, or the point at which amino acid is having no charge. Uh, and below that the amino acid will be having a positive charge, whereas above that amino acid will be having a negative charge. So depending upon the pH, that's the reason we talk about enzymes, proteins, everything, and we talk about their pH stability. So and we also talk about their functionality depending upon the pH, right? So this is the reason basically. So every amino acid has a different charge at different specific pH. So even if you change that specific pH, it can either go to neutral charge, or a negative charge, or a positive charge. So depending upon that, that's the reason each and every enzyme or a protein functions properly at specific pH, because of this specific nature of amino acid. So how those amino acids are bound together in order to make a protein? So what is a protein? Protein is nothing but made up of amino acids. So if there are n number of amino acids together, then only a protein will be made. So but how does those one amino acid joins with the other amino acid? That specific bond is called as peptide bond. So this bond you can see is, so one amino acid's carboxyl group and other amino acids amino group joins together, and water molecule is the one which goes out. So this specific H2O will go out, and the amino group and carboxyl group will form a bond with each other, which is called as peptide bond. So here the configuration by which they are binding together is, there is a form, there are two different forms basically, cis form and trans form. So this is a trans form, indicating that there will be no much of an hindrance. So cis form is the one which like they are at the same side, whereas trans form is the one which they the bond in between which they are formed, the other bond is at a opposite side. So this is called as trans configuration. This is called as cis configuration. So amino acids are generally bond with each other in a trans configuration. So if this O was at the same direction above, it would have been cis configuration, and there will be much more steric hindrance happening. So in order to make peptide bond stable, generally the peptide bond is formed in a trans configuration. And this peptide bond also has one of the property called as resonance structuring, and that's the reason it has peptide bonds are much more stable. so so this double bond is not stable enough, so this double bond and this double bond, they switch with each other, which is called as resonance, and because of this specific nature, peptide bonds are much more stable. So this was the main basics of what amino acids are and certain properties of amino acids. And along with that, there was one of the experiment to prove that amino acids are the one which will make a protein. So Anfinson was one of the scientists who tried to prove the relation between amino acid sequence and its configuration. Sorry, on its conformation. Uh, so he did one specific experiment which deals with, if one protein is folded good enough, how can we denature that and how can we refold that. and depending upon that, he basically tried to prove that amino acid sequence is the one which determines the conformation of a protein, and if amino acid sequence is different, the conformation will differ as well. So the protein he took was ribonuclease A. That's one of the protein, and he tried certain denaturants and like urea guanidine as well as beta mercaptoethanol, and tried to check whether this protein is getting denatured. And he also tried to refold it by removing this denaturants. So that was the experiment, and he was trying to prove that amino acid sequence is the one which will be responsible for protein conformation. So what does this denaturants do is, so one of it is urea, and the other is guanidium chloride. So they effectively disrupt the non-covalent bonds of protein. So the whole protein, like amino acids, will form the whole protein, and if they having certain non-covalent bonds, that will be get disrupted using urea or guanidine chloride, or else there is beta mercaptanoethanol as well. What this specific denaturant does is it breaks this disulfide bonds. So if a protein is having a disulfide bond, this beta mercaptanoethanol will try and cleave this specific bond, and those will be separated basically. So he did different experiment. In one experiment so this is the native ribonuclease A, which has 4 disulfide bonds. So you can see this specific numbers. So 26 amino acid is bonded with 84. So can you all tell me which amino acids have sulfur bonds? So these are disulfide bonds. So there are 2 amino acids which has sulfur with them. What are those?
Methionine.
Methionine and
Cysteine.
methionine and cysteine.
Methionine
Yes.
and cysteine.
Yeah, you all are right. So methionine and cysteine, majorly these disulfide bonds are with cysteine itself. Methionine is generally the start codon, and it the major one of the major function of methionine is starting the expression of one of the protein. But cysteine is the one majorly deals with this disulfide bonds. So this is the structure of ribonuclease A, and this is forming certain disulfide bonds. But when the denaturants were provided, like added in this specific protein, those as beta mercaptanoethanol is also present, it broke all of those disulfide bonds, and this specific ribonuclease didn't have any specific enzyme activity, whereas this is the native protein which had certain activity. So he tried to do with 6 molar urea as well. So here there is 8 molar urea. He tried to reduce the concentration of the urea and tried to check whether if there is some difference in the amino acid activities or the protein activity or the denaturation of those specific protein. So he tried to do that as well. Still, the disulfide bonds were broken, and it was denatured ribonuclease A was uh, obtained, and he also did the renaturation of it. How did he do the renaturation? He basically removed urea and beta mercaptanoethanol from that specific solution, and he observed that the protein was able to renature itself and form the bond, the native structure again. So how do you think this urea and beta mercaptanoethanol can be removed? Any idea? If there is a solution, in that solution there are certain denaturants as well as a protein, and you want to remove denaturants only and not the protein, how will you remove that? I think the seniors who all are present in the class can answer this. Might not be the BTech students or the 12th grade student. How do you think we can remove that, the specific denaturants from a solution? If not that, in general, if there is a certain few mixture of a compounds and you want to remove only one compound out of that, how can you do that? Like there are different ways of it, but in generally in terms of proteins or shedding protein, there is one method which is generally used for removing certain stuffs outside and keeping certain stuffs inside. What is that method called as? No idea?
Dialysis.
Yes, you're right. Yeah, so basically he in this experiment itself he tried to remove that urea and beta mercaptanoethanol using dialysis, and that's how it got removed, and uh, the protein was renatured. So in this specific, he also tried to do by adding trace amount of beta mercaptanoethanol to scrambled ribonuclease. So what happened with this is, so if you see this 26 needs to be bound with 84 and 40 needs to be bound with 95 cysteine. So this specific binding will only help the ribonuclease to function properly. Whereas if the bonding is not properly there, so we need to, I think you need to mute yourself, Tim, Tim Jose. Yeah. Okay. So these specific amino acid needs to be like the disulfide bond needs to be broken, and it needs to be formed in a specific manner so that it gives certain activity. So what and Finson done was he added trace amounts of beta mercaptan ethanol in order to degrade them, and then removed it so that it formed its native structure and it gained its activity again. So these were few experiments he tried to do and tried to prove that amino acid sequence is the one which will enable a specific conformation and enable its specific function. So urea and beta mercaptan is removed by dialysis, as you all answered, and denatured ribonuclease regained the activity after the removal, and enzyme refolded in its to active form. That's the reason it is giving activity, and the sulfidyl groups became oxidized by air. So these were something related to introduction of amino acid in part one lecture. Whatever sir taught in that. Uh, I hope everything is clear till here so that we can go to introduction of proteins. Yes. Am I going fast or this is fine, or am I going to or do I need to go much more faster because you already know about these basics? Can I rush through it, or should I go with the same speed, or should I go a little slower? As this lecture is basically mostly basic lecture, so you might be knowing all of this stuffs already. So same speed would be good. I'm already rushing a bit as you already know all of these stuffs. Or should I go a little slow? Like Pinky says, same speed. What about others? Yes. Okay, good. So this is fine enough. So we'll go with the same speed. As we are having 2 hours only, we need to also cover certain questions. So that's the reason I was asking. Yeah. So introduction to proteins. So what is a protein? Basically, proteins are made up of amino acids itself. So this different color dots represented here are amino acids. So if they are bound together, the bond between them is called as, so these are peptide bonds. And if they are gaining certain structure, they are called a secondary structure. And if there are more complex binding happening like disulfide bonds, hydrogen bonds, etc., then it forms a tertiary structure. And if one tertiary structure and other tertiary structures come together, like certain proteins require two polypeptide chains to form the whole protein. So if that's the case, then that comes under quaternary structure. So this is just an overview of proteins. We'll go a bit in detail about each of the structures. So proteins and their properties. So what all things will be discussed in this specific lecture? Like part of the lecture is about introduction of proteins. We'll be dealing with proteins and the properties, different structural levels, primary, secondary, tertiary, quaternary, etc. And if the protein is not specifically present or absent or misfolded, anything happens, then what kind of diseases is possible with that? So what is a protein? It is derived from a word called as proteose, and what does it mean? Is a linear polymer built of monomers. So here monomers are nothing but amino acids, and the it is the most versatile macromolecule in the living system, and it plays very important roles in uh, different functions basically, and it is very important in general because ultimately whatever gene you are having, it is functionally expressed as a protein only. So if If your gene is accurate, your protein will be accurate. Still, there are certain different modifications happen with the protein itself in order to make it functional, and so it's basically more important to us. And it has different functions. It has functions in signaling, it has functions in transport of one molecule from one place to another place. It has functions in catalysis, it has functions in movement, it has functions in structure, regulation, etc. It has n number of properties basically. So these are certain examples of proteins like hemoglobin, insulin, keratin, etc. And each of those proteins have performed different kind of functions basically. So hemoglobin is the one is a protein present in our blood, and it is responsible for the transport of oxygen. So the major role of hemoglobin is transporting oxygen from one place to another place. So that's transport. Whereas insulin is the one which acts as a messenger. So if there is a sugar level inside our blood, so insulin will act as a messenger, and it will try and uh, cleave those sugar molecules into, so that it gets degraded and there is no blood sugar level increase in the blood itself. So it acts as a messenger. Whereas keratin is a protein which is present in our nails, in our hairs, etc., which helps in structural support. Whereas actin is a protein which helps in movement. So inside the cell itself or inside the muscle itself, uh, it helps in movement. So that protein gives us strength of movement. In terms of enzymes, like DNA polymerase helps in DNA replication. There are certain proteins like antibodies like IgG which helps in, which is a property, which has property in our immune system. It helps in defense, etc. So there are n number of proteins with n number of different functions. uh, so all these are come under a broad umbrella called as proteins, and one of the most abundant protein in nature is ribosco. Do you all know where is this protein found, in which cycle, etc.? Yes. Where is ribosco found? What does it do? No idea. You have a doubt regarding what? Sashi Rekha. The questions will be asked like MCQs, or I think we can discuss that later also. There will be a written form as well. There will be MCQs. Majority of them will be MCQs, but I think there is a written form of exam as well. I need to clear that with NPTEL. Like your assignments are having MCQs only, but yeah. So in terms of primary structure, so each amino acid joins together. If the sequence itself is called as the primary structure, and this amino acid has two terminals basically. The first amino acid has N terminal and the last amino acid has carboxyl terminal, because the bond which is formed between two amino acid is the carboxyl group of the first amino acid and amino group of the second amino acid. That's how a peptide bond is formed. So these are peptide bonds, and the N terminal end is the first amino acid, and the carboxyl group end is the last amino acid. That's how specific sequence is. So this is called as the primary structure of protein. What does it do? The primary structure is essential for elucidation of its mechanism of action. It helps in analyzing the 3D structure of proteins, and it helps in and this specific each, if there is a change in one amino acid also, the function might differ, and it can produce abnormal protein, or it can lead to abnormality. And this basically, this specific sequence will also try and tell us an evolutionary history of the protein as well. If you all have done blast and stuff, it will be easy to understand that those things. And in terms of the secondary structure, so those proteins itself are aligned. So this is the first amino group with the fourth carboxyl group. They form bonds with each other, and they form a structure like this, which looks like an helix. So it's called as alpha helical structure. And if there is a bond between such parallel amino acids or anti-parallel ways, so there are beta plated states are in two different ways, parallel and anti-parallel. If there is a, if such kind of bondings are happening, hydrogen bonds, if you can see here, if that specific bonding leads to a beta plated structure. So if if we just look at the structure sequence, it is called as primary. And if certain kinds of hydrogen bonds are happening in a specific manner, those are called a secondary structure of the protein. So how does this secondary, in terms of alpha helix, it is the carboxyl group of first amino acid and the amino group of the fourth amino acid. So this is 1 2 3. So this amino acids carboxyl group, then 2 3 4th amino acids amino group. So that's how if these two are joined together, like in this manner, if you can see here, so this is the first, there will be second backside, third here, and this will be the fourth one. So if then it will form alpha helical structure. So that's how it is. So this is the first one and fourth one, and it has a certain uh, rotating angle. And basically we can say 3.6 6 residues exactly. It's not exactly the fourth one, but it's the fourth amino acid basically, and it forms certain phi and psi angle. So what are this phi and psi angle? We'll deal with it in the coming slides. So in terms of beta sheets, or so this is then another name of beta sheets itself, and they can be parallel or anti-parallel, indicating that if the amino acid sequence are going in this direction, in the same direction, they are called as parallel beta sheeted structure. And if it is going in opposite direction, one is going here and one is going at the opposite direction, and that's how the structure. If the binding is happening between these two, then they are called as anti-parallel beta sheets. And these are turns and loops. So this is a loop here, and these are turn here. So the turns and loops are formed in terms of beta sheeted structures. So those are more elaborated structures. So loops are generally rigid, and turns and loops are on the surface basically. So if a protein is folded, those loops and turns are generally on the surface, and these structures are embedded inside basically of the whole protein. So this is the general difference between them. So in terms of alpha helix, those are tightly coiled. Similarly, is in terms of beta sheet, it is fully extended. You can clearly see that. So this is tightly coiled with within the structure itself, whereas these are extended clearly. So that's how it is. And those distance is 1.5 Armstrong in terms of alpha helix, and this is 3.5 Armstrong. So there is hydrogen bonds forming in terms of alpha helix and beta sheets, which helps in forming basically the secondary structure of the protein. And there are certain examples of it. So phi and psi angle are these angles. So if the amino group of the first amino, the angle between the alpha carbon or chiral carbon with amino group, this specific angle is called as phi angle, and the angle between the carboxyl group and the C alpha carbon. So this specific angle formed by this specific bond is called as phi angle. So these are psi and phi angles. So this specifically, so one of the scientists called as Ramchandran has made a plot of those using those angles. So whatever these angles are phi and psi. So these, the one in red are the most preferred angles. So these phi angles, if this is a phi angle minus 90, then generally the, so whatever this might be around minus 80 or something. So this specific angles will be preferred for a stable structure for right-handed alpha helix. Similarly, those specific angles are stay. I didn't get you. Hello, voice is breaking. Can you hear me now properly? Yes. So this is the Ramachandran plot, basically. So these are certain specific angles which are preferred for a specific protein to get folded. So these phi and psi angles were studied by one of that specific scientist, and he made this specific plot saying that these are preferred angles for a protein structure to be stable. So in terms of tertiary structure, it is a three-dimensional compactly folded structure, and it's the overall organization of those secondary structure, both alpha secondary as well as certain covalent bonds, certain van der Waals forces, etc. All types of electrostatic interaction, every kind of interaction, all those disulfide bonds. If everything is there, then it forms a tertiary structure. So here we are just dealing with hydrogen bonds at specific method. So if this is the method, it's called as beta sheets, and if this is the specific method, it's called as alpha helical structure. That's how it gets folded basically. So this is hydrogen bonds deal with secondary structure, and if we are considering all types of interaction, then it forms the tertiary structure. So if it was there in the first slide itself. So this is just the secondary structure, the helical structure or a pleated structure, and all those disulfide bond interaction, electrostatic interaction, van der Waals forces, etc. All those interactions, if they come together, so this specific structure will be much more compactly folded, and that specific structure is called as tertiary structure of the protein. Yeah. So then in terms of quaternary, it's so these quaternary structures are only for those proteins which require two different polypeptides to make one specific protein. Like insulin, there are two polypeptides for insulin. There are four polypeptides for hemoglobin. So for those kind of proteins, we would need quaternary structure, and so each polypeptide will have its own tertiary structure, and if they are bound together to make a functional protein, that will be the quaternary structure of the protein. So this is one of the example of hemoglobin which sir gave. Like fetal hemoglobin is slightly different than that of the adult hemoglobin. So fetal has alpha and zeta if I'm not wrong, and whereas we are having alpha and beta structures. So this alpha, these are four polypeptides basically: alpha 1, alpha 2, beta 1, and beta 2, and so this each is having their own tertiary structure, and if they are bound together, it forms the tertiary structure. And sir did explain a bit about hemoglobin, how does it function, or why it has a red color, because of that uh, ion present in its structure. So this specific protoporphyrin is present in the middle of the structure, and that's what enables all polypeptides to come together and form the whole structure. And along with that, we tried to compare hemoglobin with myoglobin. It does have a similar structure, but they are different. And so how can we determine a specific structure of a protein? By using X-ray crystallography technique, a protein will be crystallized, and that because of that crystal, those X-rays will be diffracted in a different direction depending upon how the crystal is of that specific protein, and that crystal will give certain graphs like this, and that will help us to determine the structure of the protein. So it's a bit complicated process though, but that's the overview how X-ray crystallography works and how a protein structure is determined. So I did talk about the oxygen binding capacity of myoglobin and how exactly it is calculated. So they did one of the experiment, and there is at a point where there is a saturation, it cannot bind to more oxygen. So that's the capacity of oxygen by which myoglobin can bind to oxygen. So in terms of this lecture, in terms of this part of the lecture, we talked about how primary structure is, secondary structure is, tertiary and quaternary are, and a bit of okay. Along with that, as I said, we'll be talking about the diseases if the proteins are not folded properly, or if there is a change in one amino acid. So, for example, hemoglobin generally has glutamine at one of the side, but if that is changed to valine, just by replacement of one specific nucleotide from A to T. So GAG codes for glutamine and GTG codes for valine. So if A is replaced by T because of the change of one specific nucleotide, there is a change in the amino acid, and the change in one amino acid enables, like, leads to this specific disease called as sickle cell anemia. Similarly, this is the normal sequence which has a splice site. If that is changed to another site, another splice site, it leads to beta thalassemia. So change in, even there is a change in one specific nucleotide or one amino acid, can lead to different diseases. Similarly, so did talk about Parkinson and Alzheimer's as well, uh, as well as lathrism, which is caused because of lysine, and it forms a different structure because of lysyl oxidase, and because of that specific different structural formation, the protein is not folded properly, and this specific disease is caused. So that's was it is about introduction to proteins, and if you all want a 5 minutes break, we can take that, and we can go ahead with part 3 of folding and misfolding. Yes, so it's now 3:55, so we can start at 4 o'clock in like next 4 or 5 minutes. अरे मला किती कव्हर करायचं आहे अजून, 2 तासात यायचं आहे. असंच होतं. टीचर झाल्यावरती समजतं, तेव्हा तुम्हाला एवढं कव्हर करायचं असतं ना, तेव्हा रिअलाइज होतो बाई ते लोक तसं का करायचे म्हणून. Where are you all from, by the way? Like only Lekha answered as she is from Tamil Nadu. You all are pursuing your degree at in which college or where are you working? Okay, others? Okay, we'll start with the lecture again. So what is so as we talked about the structure of the protein, so it requires certain way of folding by forming certain hydrogen bonds or disulfide bonds, etc. So only then a protein will be functional. So how does so in terms how folding or misfolding can affect the protein function and how that can cause. We already talked about certain diseases as well, but a bit more about that related to thermodynamics and how a protein folding can be made easier. What are those molecules called as for making proteins to get folded? So those are chaperones, and this we already talked about, like protein misfolding and diseases. A bit of it we talked about in the earlier slides too. So generally, in terms of protein folding, uh, polar amino acids tend to gather on the outside of the protein because if you generally if you see most of the proteins are soluble in water, right? So why is that? Because all those amino acids facing outside are polar in nature, and the non-polar amino acids are buried inside. So if a protein is folded properly, some amino acids are facing above, like not facing above, basically they are exposed externally, and that's what makes protein soluble. So we already discussed about it. So this is a good uh, uh, non-polar or polar positive negative. So this gives the whole overview of amino acids. So generally amino acids are coded by either three-letter word or a single letter symbol. So you can also remember them or just go through that once. So, in terms of thermodynamically, how a protein is getting folded, because so in terms of in the folding process itself, the overall increase in an entropy which drives the folding process. So these are polar side chains, the one in brown, and the one in green are non-polar ones. So generally, this is how the protein tries to fold itself. Why? Because it wants to expose those hydrogen bond interactions to happen with the water molecule, and that's the reason it tries to fold it like this, and it is, whereas hydrophobic residues are inside. So hydrophobic in the sense non-polar, and hydrogen or the one which are polar in nature, those are polar side chains. So that's how the generally the folding happens in aqueous environment. And this we already discussed in terms of Anfinson experiment. The protein can be denatured by exposing to certain denaturants, and it will be whatever is the folded form, it will get into a linear form, or a primary structure is achieved again with the help of denaturant. And if we remove that, original conformation can be re-achieved. That's what was proved by Anfinson as well. And so this is how generally it is. The protein unfolded at a higher rate when denaturant is at a higher concentration. Uh, so if there is no denaturant present, the protein is not unfolded. Protein is folded properly, and if the denaturant concentration increases, the protein gets unfolded. Yeah, and generally in terms of folding, as I said, it needs to be folded at that specific amino acid, or basically those specific 16 amino acids, the one we are talking about in terms of ribonuclease A. Only those 16 needs to be found, needs to be together to form the disulfide bond. If then only that specific protein is functional. So there are many unstable conformations, even if there is certain interaction happening, those are not stable. There is only one stable conformation for one specific amino acid. So only if it gets folded in that specific manner, then only the protein will be functional and do its function. So folding is a cooperative process. In general, any protein adapts to only one conformation or a few very closely related characteristical functional conformation, which is called as the native state. So these are called as the native state, and the one which is not stable, though those n which will be getting degraded, and the body itself will try to form the completely folded stable protein. So this is how the process of folding is: unfolded state, this is partially folded state, and this is folded to a native state, completely folded. So this is the stage where it is partially folded, this is completely unfolded, and this is folded. That's what I was talking about in the earlier slide. Yeah. So in terms of thermodynamics, so folding of proteins into their native conformation occurs spontaneously under physiological conditions. So what do you mean by physiological condition? In the sense, whatever is the functional condition, logical means functional, and at that specific conditions, it will automatically form its native conformation, and it is majorly dictated by the primary structure of the protein. If there is a change in one single amino acid, that structure might not form the way it's supposed to be formed. So if the primary structure, or the basically the sequence of the protein is accurate enough, then only it will form the secondary, tertiary, and the whole native conformation is achieved. And in terms of thermodynamic itself, the entropy of that specific amino acid keeps on decreasing as it goes to a stabilized form. So entropy is something which is not stable, right? It is major degree of randomness basically, and so that randomness is in the primary structure, or the unfolded one. As it tries to fold, the entropy energy decreases completely, and it does form a stable structure then. So this is the native, primary structure, and those water molecules are tried to expose, and it is trying to form the helical structure, and that's how the free energy decreases uh, in a attempt to achieve the stable structure of a protein. So in terms of those protein itself, these conformations are happening with the help of certain enzymes or certain proteins like DNA J, DNA K, phosphate, group E. So the GRPE basically. So these are DNA J, DNA K. These are actually helping the specific unfolded protein to form the folded protein. So how does that happen? So initially DNA J, DNA K binds, and then ATP comes into action, and then DNSJ goes out. DNSK goes out of it, and along with releasing two phosphate bonds, and DNSJ still binds to the protein itself, and with the help of this GRPE, uh, and DNAJ, these together along with as 2pi are two phosphate bonds are eliminated from these two ATPs, so there is ADP present. So these three together will help to form a partially folded protein, and then with the help of GROEL system, it is trying to form the whole completed structure. So these all things are actually called as chaperones. So these chaperones are the one which actually help the structure, the protein to get folded into its native conformation. So that's how it is. So initially, these DNA J and DNA K binds with the primary structure of the protein. Then this specific GRPE protein comes into action, which cleaves the DNA K here, and that is removed, and ATP is also used up, wherein two phosphate bonds are released, and this DNAK is regenerated in the next round. That's the other process, but this specific process helps in partial folding of the protein, and then this specific system will help the protein to get completely folded. So in terms of misfolding, protein folds into a single energetically favored conformation specified by its amino acid sequence itself, and it may fold into alternative 3D structure also. Why it can fold into alternative 3D structure? That happens because of mutations and inappropriate covalent modifications, and this will lead to different diseases basically. So if this is uh, generally correctly folded, will be dissolved properly, and if it is not folded properly, or if chaperones are not present properly, which helps in which will not help the protein to get folded correctly, it will form protein aggregation if it is not correctly folded. So the misfolded protein generally gets accumulated, and the proteolytic fragments result into few degenerative diseases. There are a number of different diseases, and Sir gave few examples of it, and it is characterized by presence of insoluble protein. So how does an insoluble protein is formed? Because it is unfolded. Then only it is an it is an info uh, like not properly folded protein. That's the reason it forms. It goes into insoluble fraction, and it can form. It can go in certain organs and form certain diseases. So, sir gave these certain diseases options. We did talk about few of them in the earlier, but there are few others as well, like Huntington's disease, cystic fibrosis, pulmonary emphysema, etc. So all these are different diseases caused because of change in one specific protein sequence, or so basically these diseases are caused because the proteins are not folded accurately as they are supposed to be folded because of certain mutations in the sequence itself. So that was about protein folding, and uh, later in the lecture, sir did discuss about protein purification. There are different techniques to purify a protein, and this is one of the way, but we'll talk a bit in detail. So these are the few techniques sir talked about: gel filtration, ion exchange, affinity, HPLC, and reverse phase chromatography. So these are few chromatographic methods sir talked about, which helps in protein purification. So, what does this chromatography technique deals with? It deals with uh, differential solubility of proteins, so that can, so depending upon the solubility, we can separate the protein. Depending upon the size of the protein, we can separate. Depending upon the charge, we can separate. Depending upon the affinity, so we can choose any of the function of a protein, and that function will basically help us to separate as well. And there are different ways, different methods for each specific function. So depending upon charge, there is isoelectric focusing, or isoelectric chromatography. Similarly, depending upon affinity, the interaction between two of one amino acid with the other, that will help in affinity chromatography, and that's how we will separate. Depending upon gel filtration chromatography, depending upon size, or we can do SDS page, etc. So those all comes under size, uses the property of size of a protein and then separates it. And depending upon solubility, like salting it, salting in, salting out, ammonium sulfate purification, etc. All those comes under. So we can use a specific property of a protein, and using that property, we can try and separate the proteins. And depending upon that, there are different methods which are already established. So what are the major things chromatography is involved in? Like has is the sample addition. Once the sample is added, that it has a mobile phase, it has a stationary phase, and once all those are separated, we need to detect them, whether how they are separated or which collection fractions uh, our desired protein is present. So we need to have a detector, or there are different ways of detection, and accordingly we need to know that specific protein how it can be detected. Similarly, chromatography requires a matrix. Basically, this stationary phase will be depending upon the matrix. It can be either bead shaped. It depends upon the size. It depends upon the porosity. It depends upon the charge, etc. So this stationary phase will talk about all of these stuffs. So these are the major things involved in a chromatographic assembly. So for example, in terms of gel filtration chromatography, generally it is beads, and it is generally we can either have a chemical group attached to it, or it can be without the chemical group as well. And those interaction with the column matrix is an important feature of chromatography. Depending upon those interaction only, a desired protein will get separated. So one of the way of separating protein is gel filtration, which uses a property of size of a protein. Depending upon the size of the protein, the uh, proteins will be getting separated. So smaller the size, it will be retained longer in the gel filtration system, and larger proteins will elute fast. That's how the this technique works. So if protein is larger in size, it will be coming out in the earlier fractions, whereas if it is smaller in size, it will be coming out in the later fractions which we are collecting. So if you can see, this is how an example of protein separation in terms of gel filtration is. So your sample is having two types of proteins here. One is red and one is green. So the red one is larger in size and green is smaller in size. So what is happening is red is going away from those specific beads. It is not going inside the beads here, whereas the larger protein is going from outside of those beads. That's the reason it is able to flow in a much more faster manner. It is coming out earlier, whereas smaller proteins is going inside the beads because of the pore size of the beads. And because of the excessive traveling inside those beads, it is eluting out slower. So that's what the principle is in terms of gel filtration chromatography. So the larger particles or the larger proteins will get eluted first, and the smaller will be getting eluted later. So once these fractions are collected, we need to analyze them. So one of the way of analyzing is UV spectrophotometer, and we can just, as I said, we can just check the protein concentration at 280 nanometer. We can do that way, or else there are different methods also for quantification, like Bradford assay, etc. And if we can basically add that specific dye and then check at specific nanometer and check whether there is a protein present in your collected fraction. So that's how you can detect it. So once you check all those fractions, you will try and understand. So earlier ones were higher molecular weight one, and the later ones which had certain proteins which are of lower weight proteins. Similarly, there is ion exchange chromatography as well. In terms of this, this separation is using the property of charge of a protein. So it can be either positive charge or negative charge, depending upon that different matrix are available. So here the solid or the stationary phase can be cellulose or agarose, which is sorry the stationary phase can have carboxymethyl group or else diethyl aminoethyl group, which so this DEAE has a positive charge. So negative proteins can be separated using this as a matrix, whereas this has a negative charge. So positively charged proteins can be separated using this matrix, and they are attached to a matrix called as agarose. So this uh, are attached with a group called as carboxymethyl, and that will enable binding of those positively charged proteins, here the negatively charged proteins, and that's how separation will happen. Basically, after attachment, we actually add a elution buffer as well, and that will help in elution of the those specific uh, selected desired amino acids, desired proteins. So this is also similar, but here the beads are having certain charge. So here it's an example of negatively charged proteins, negatively charged beads, like they have used carboxymethyl as an example. So here the beads are negatively charged. So positively charged proteins will be getting interacted with these beads, and because of that interaction, they are going slower. whereas the negatively charged proteins will repel and they will not get interacted with those beads and they will elute out faster. So that's how ion exchange chromatography works. Similarly, then we'll have to detect it in one of the method like here, similarly, after the collection of fractions. Similarly, in terms of affinity chromatography, it depends upon how your specific protein is attracted to one of the ligand attached to the matrix. So based on affinity of a protein as compared to that of other molecule, that's how separation will happen here. And here, one of the ways metal chelation, like nickel column is used, and if your protein is having histidine tag, histidine is generally attracted towards nickel, and uh, that's how recombinant proteins are purified using affinity chromatography technique. Uh, so this matrix beads are chemically coupled to a ligand, whereas in terms of gel filtration chromatography, those are not attached. There is no attachment of any specific ligand acid. It's only the pore size of the matrix itself. Whereas in terms of affinity, that specific beads are attached with certain ligand which will have an interaction with your desired protein. So here, so this is the ligand, the bead to the bead itself. There is a ligand attached. If you can see the one in black here, and that will have specific interaction towards specific kind of proteins only. So there are three different types of proteins here: red, blue, and green. Whereas it is getting interacted with only red colored proteins, whereas blue and yellow are getting eluted out first. So these are getting binded with each other. Then we'll add a elution buffer, which will break this specific binding, and only those specific uh, binded proteins will be getting eluted, and that's how separation happens in terms of affinity chromatography. There are different types of fusion partners available, like histidine tag requires nickel ligand. Similarly, if your protein has protein A, that can be separated using IgG, etc. So this is how different matrix can be formed. So these ligands are the one in black here. So there are different options of those ligand depending upon your protein. So this, if your protein is having histidine tag, then your ligand should be nickel. Similarly, if your protein is having protein A, your ligand should be IgG. Only those specific combination will work accordingly. And if you want to elute them out, so once this binding is happened, you need to add a elution buffer. So as an example, if there is histidine tag and nickel uh, ligand, so your elution happens with imidazole. So your elution buffer will contain imidazole, which will have higher interaction with this nickel, and your desired protein will get uh, detached from here and get eluted out. So accordingly, those functions needs to be checked, and accordingly, elution buffer needs to be made for your elution using affinity chromatography. So this is how it is. It's the same thing, affinity chromatography itself. So this is an histidine tag, and if it is binding with the nickel, it will get eluted later once the elution buffer is added. Whereas all other proteins will get eluted first, as they will not interact with the ligand. Similarly, there is one method called as HPLC, which is high performance liquid chromatography. Here, the separation is on the basis of difference in affinity for both the phases, stationary as well as affinity. Here, the interaction is with the stationary phase itself. There is not much interaction with the, there is, we cannot say there is an interaction with mobile phase as such here. It's just the buffer, but here the interaction depends upon both stationary as well as mobile phase. Depending upon that, the separation happens. It also helps in removing undesired impurities, and it for this we require concentrates the diluted samples as well. So that's how HPLC can be used. So there are different mobile phases. The stationary phase is the column itself. uh, there are different columns available with silica or else with carbon. Uh, so those is that is the stationary phase, and different mobile phase, and are added to that specific column for elution when your sample is injected, and depending upon the interaction with the mobile phase and stationary phase, your protein will get eluted out. So one of the method is reverse phase chromatography. Here, based upon the hydrophobic binding interaction. So whatever sample you are having, if it is hydrophobically, uh, it has interaction of hydrophobic. Basically, if it is non-polar in nature, it will be immobilized with the stationary phase and it will get eluted out later. Whereas if it is polar in nature, it will get eluted out first. So that's how this reverse phase chromatography is used. So this ESI is electron spray ionization. That's the later stage after separation. So here the detector is mass spectrometry itself. So for mass spec, basically your sample needs to be ionized. So once the separation is happening, so this technique is basically called as LCMS. In HPLC, your sample is getting separated, and in MS, initially you need to ionize your sample before going towards the MS. So that ionization is happening with ESI, which is electron spray ionization, and your sample gets ionized, and that because of that ionization, MS can detect what specific mass is present in the sample. Then there is SCX as well, which is called a strong cation exchange resin, which also comes under ion exchange chromatography itself. It's similar to that of ion exchange chromatography, whereas silica-based cation exchange stationary phase is used, and with the help of that, anions can be separated using this technique. Then the final lecture or the final part of the lecture was the introduction to proteomics. So, what is proteomics basically? Can you all define in general proteomics? If you all know about it, or we can go ahead. I hope you all are getting it till now. Yes? Okay. So, what is omics in general? Okay, you all don't have to call me sir as such. You can call me with my name as well. What is omics in general? Can anybody define that? Okay. So, if I just talk in a layman term, omics is basically everything you want to study about that specific uh, topic or that specific region or that's so if you are talking about proteins, so each and every point of that specific protein, each and everything concept of that specific protein will come under proteomics. So omics is basically everything. So and similarly, there is genomics as well. There is transcriptomics, there is metabolomics, etc. There are different types of omics studies. So in genomics, people study about DNA, about the gene itself. In transcriptomics, so after the DNA, if we transcribe RNA, we'll get RNA. So if you are trying to study RNA, that specific study is called as transcriptomics, and after RNA, it gets translated to protein. Then if you are trying to study about the protein, it's called as proteomics. And along with that, there are certain metabolites present inside our body. If you are trying to, like, metabolites in the sense all those except that of protein, carbohydrates, lipids, etc. And if you are trying to study about that, then it is called as metabolomics. Okay, before going this, I think I need to talk about this first, and then we'll talk about proteomics in general. So these are certain components of omics. So in terms of genomics, you are trying to study about the DNA, each and everything about DNA, how DNA is getting replicated, what is the sequence and how one specific sequence can affect one thing, etc. each and everything about the gene. Epigenomics is depending upon the environment, the gene changes depending upon this different histone variations as well as methylation etc. of the gene. That is called as epigenomics if you are trying to study that. So basically genomics and epigenomics correlate with each other a bit, and uh, transcriptomics is after the gene once it gets transcribed, and if you are only studying about this part it's called as transcriptomics, and if you are after that once it get translated, if you are studying about protein it's called as proteomics. And so these proteins to get so there are certain metabolites, these metabolites gets converted to one form to another form or whatever with the help of these proteins, those are called as metabolites. So these are metabolomics. So depending upon that, there are different omics components, and here we will be just dealing with the proteomics part. So so proteomics, as I said, it is everything about protein, how a protein is analyzed, characterized, the functions, etc. Everything it comes under proteomics umbrella. And so if so here in terms of different steps involved in terms of proteome analysis is we'll do protein extraction, we'll try and separate that. So we did talk about separation of protein, that is purification of protein. Once that is purified, how will you identify that it's the same protein which you wanted to separate? And if once it is identified using mass spec etc., then you need to characterize that exactly. If there is an unknown protein which has been separated and you want to identify that and characterize that, all those are the steps which are involved in terms of proteome analysis. So how proteomics has been evolved from 1975 till now, even ahead now. So initially there was only gel based proteomics. This is called as 2D gel electrophoresis or DIGE, differential gel electrophoresis. So around till 1990, those techniques were used in order to study a proteome or a proteome technique. Then later, since 90 after 1990s, different algorithms came into picture, and proteomics uh, was boosted because of uh, nucleotide sequencing being happening. So we did know what specific protein is has been coded by which specific gene sequence, because we came to know about nucleotide sequencing. That has, so this basically might be helping genomics, but this actually helped proteomics study as well, because now we know the sequence of it. And because of the mass spec introduction, we did know about what exactly mass of a protein is, and basically that uh, enabled the analysis of proteins in a much more efficient way. So after the separation, this LC is dealing with separation of proteins, and MSMS is dealing with uh, the fragmentation of that specific protein. So certain proteins are very complex, right? So we need to fragment them and then analyze it. So once it is getting analyzed, fragmented, we can analyze them. So that complex protein can also be analyzed. So that's how LC-MS-MS work. And there are chip based approaches as well, wherein uh, in the chip itself there will be certain immobilized stuffs, and that immobilization will actually help the protein to get interacted and help in analysis etc. So there are different chip based approaches, different MSMS based approaches depending upon the detectors, and that's how proteomics got evolved. But although will do talk about MS in this course, but majorly it will be about gel based proteomics and not about uh, MS based or LC based proteomics. So proteomics can be divided into two, either abundance, you just need to identify, and quantity, understand the or quantify it, where else we can also try and check the function. So this chip based approaches deals with functions, expression, assays, etc., whereas MS is just deals with quantification. It will not tell you about that specific if it is functional or not. So this will not tell you about that. This will just tell you about if there is a protein in one certain sample, how much concentrated it is, if we have standards available. Then only we'll try to know, like, understand what's the quantity of it. And we can identify it as well depending upon the retention time. So when there is a separation happening in terms of LC or HPLC, we call it as at what time it is coming out, the elution time. That specific time is called as retention time. So each specific protein will have its own property to come out. So if it is coming out at one specific time, it will, by using one specific method in the sense having one specific mobile phase, having one specific stationary phase, if at one specific concentration, and if it is coming out around 5 minutes or 10 minutes, it will always come out at that specific minute itself if you are using same method. So that specific time is called as retention time. So depending upon that, we can identify it as well as quantify it using LC-MS-MS based approach. But if you want to understand the expression, assay, etc., you need chip based approaches, etc., and even other methods are available for it. But we'll be majorly dealing with gel based itself. So this is light blurry, but this is an MS approach, uh, which we can talk in our later lectures as well. So in this specific lecture, sir actually gave a broad overview of everything in general. So DNA has exons and introns with it. So introns are the one which are non-functional in nature, and we need to remove that. So transcription happens with the whole introns exons both together. But when in the transcriptomic study itself, there is alternative splicing happening, whereas introns are removed out and only exons come together, and this specific RNA can form three different types of proteins depending upon what kind of exons are coming together. If 135 is coming together, there will be one different protein which will be formed. So this is the, you can also call this as a central dogma of our body or of a cell, whereas DNA is transcribed to RNA, and RNA goes through alternative splicing to form different types of proteins. And then it gets translated to form that specific protein, and that protein can undergo different modification which is called as post translational modification. So this is translation. So once the translation is happening, if we want to modify that, that's called as post translational modification. And that modification are different types. These are a few ways of it: phosphorylation, hydroxylation, glycosylation, methylation. Basically that specific group will be added, phosphate group is added, or a glycosyl group is added, or a methyl group is added, something like that. So those are post translational modification happening with the protein to make it much more accurately functional. And there are certain methods to detect the protein. One of the method is Bradford method, wherein a dye called as Comasi brilliant blue is used, and that dye comes in contact with the protein, changes its color, and once that color can be, it becomes blue in color basically. But here, although the description or display is green, but generally this interaction makes the protein to get converted into bluish color, and that is determined at 595 nanometer. So that can be one method for determining the protein concentration. The separation depending upon size, one of the method is SDS page as well, where the separation is depending upon size. The larger molecules will be uh, uh, separated at a, or moved at a very slower rate under the current, whereas the smaller molecules move faster, and that's how we can get the separation of the proteins. Similarly, in terms of isoelectric focusing, we load the sample in between, and whichever proteins are having negatively charged will go towards the positive direction, and which are positively charged will go towards the negative direction in order to find their isoelectric point. So it is a point at which there is no charge. So A and B here, the A and B option which are given here. So the one which are going above, they were initially positively charged, they were going towards the negative and achieving the neutral side, and those proteins which were negatively charged, they tried to go towards the lower pH and get their stable isoelectric point, and that's how the separation happened in terms of isoelectric focusing. So in this whole lecture, he tried to give overview of all the steps. So he did also give overview of 2D gel electrophoresis, wherein initially once the samples are prepared, the initial separation is on the basis of IEF, then the later separation is on the basis of SDSP. So if you are doing two together, then it's called as two-dimensional gel electrophoresis. It's the same method, like you are doing IEF first, isoelectric focusing first, and then you are doing SDS page. So if you are doing two together, then it is called as 2D gel electrophoresis. There is one method called as dye as well, which is differential gel electrophoresis. So difference here is basis on the basis of dye itself. So there is a control one which you will treat with one specific dye, and treated one which you will treat with one specific dye. Then the pooled one, like which are having both together, that is the pooled sample, you'll add a different dye. Then you will mix all of these three, and then uh, do this 2D gel electrophoresis after giving them different dye basically. So because of this different dye, once they are separated, when you do the image analysis, you will try and understand uh, from which sample, from which control and treated. So in the mixed sample, you will understand whichever are. So here the color depiction is not similar, but what happens is you will try and understand what all kinds of proteins are different in the treated sample and what are different in control samples, and accordingly, so this intensity, this uh, larger round and smaller round talks tells you about the intensity of those proteins, and accordingly you will try and determine uh, which specific protein is abundantly present in the treated state or in the control state by comparing it with the pooled internal standard. So that's how this specific technique works. This is about the whole uh, week one lecture which sir talked about. He talked about all of this. Initially, he talked about introducing proteins, amino acids, did talk a bit about folding, misfolding technique, purification techniques, and a bit of proteomics. So this proteomics study will be much more easy to understand in these two techniques, these two lectures, uh, uh, two-dimensional gel electrophoresis and differential gel electrophoresis. How this technique will try and help you to understand the proteomics approach. Basically, all this will come under proteomics itself, but as we are talking about gel based itself, so that's the reason these are two gel based techniques, two dimensional and differential gel electrophoresis. Today I just gave an overview of it, like how does 2D gel electrophoresis work and how does DIGE work, but we'll go much more in detail in the coming lectures. But this is just an overview of what all you will be studying in this course, and for studying this course in a, like, to understand this course in a easier way, that's the reason we spend more time in understanding the basics of it, what a protein and how purification techniques are. So this will help you all to understand the gel based proteomics approach study. Yeah, if you all are having any doubts, we can take that, or else we can do a few questions uh, related to the topic. Oh my god, my mic isn't working. Why is that so? Hello, am I audible? Since when I was not audible though? Like 2 3 minutes back? Okay, so so when I was going through all the slides, just revising everything, so that's what Sir deal with in this specific week's lecture. I hope everything got revised, whatever sir taught. So I just tried to revise it everything with in a lesser amount of time instead of taking 3 hours. So I tried to do it as quick as possible. I did go a little faster, but I hope it was as it was basics. You people were getting it. That's what I'm thinking. I don't know whether you all got it. If yes, you all can just text that, or else we can go through the questions as well, which are easier comparatively. I'll send the PPT. I'll be so there's a way NPTEL does. So we'll be uploading the. So I think you all might be given the specific links to access. So I'll be adding the link here of the up like this recorded video as well as uh, I'll be uploading it this specific PPT. So I'll be adding the link here as well. So you can once you click those links, you will be able to get the PPT as well as the recording of this lecture. Yes. Okay. Should we go through the questions? I think I need to do it in a different way so that I can see the chat box. Okay. Can you all see this Word document? So we can do the questions. Few of the questions related to the topic which we studied. So what is the answer for first question? So which functional group are common to all standard amino acids? Amino group, hydroxyl group, sulfhydryl group, phosphate group, aldehyde group, ketone group, and methyl group. One of you have answered it as A. How about others? You can see the question. Yes, only Aditi is answering. How about others? Okay. Yes, the answer is A. So which amino acid is optically inactive? So optically inactive will be the amino acid which is not chiral in nature. It will not form mirror images. So which one is that? Glycine, alanine, leucine, proline. Is the next question. Glycine. Yes, the answer is glycine. Where's the document? Okay. The chirality of amino acid is due to what? Due to the presence of a symmetric alpha carbon or the presence of a sulfur atom or a planar peptide bond. Is it because of the peptide bond or is it because of ionic side chains? Because of what? Third one is also A. Yes, I think it's better to answer like the way Aditi is answering because that will help me to understand for which question you are answering. 3A or something like that. Okay. The primary structure of a protein refers to what? We are talking about the primary. So is it 3D conformation? Is it sequence of amino acid? Is it assembly of subunits or hydrogen bonding pattern? Yes, fourth one is B. Can you all answer this? You can take the same question and just write like the A option belongs to primary, secondary, or tertiary. Like you already said, B option belongs to primary. So A option belongs to which type of structure, C and D. So you can talk about that as well. I'll just give you the question again. So this the question itself has it belongs to tertiary though. But what are these two? C belongs to which type of structure and hydrogen bonding pattern belongs to what type of structure in terms of a protein. 4C is quaternary, yes. And how about D? D is secondary, yeah. 4A is tertiary, 4C is quaternary, 4D is secondary. Yes, you're right. Let's see the next question. Which bond is not involved in stabilizing protein tertiary structure? Disulfide bond, hydrogen bond, peptide bond, hydrophobic interactions. So here you are talking about tertiary structure which is not involved. 5A. Okay. Disulfide bond is not involved in stabilizing protein tertiary structure. We are talking about stabilizing the protein tertiary structure. Okay, 5A, 5D. So people are confused between A and D. The answer is C actually. So this peptide bond deals with the primary structure, right? So we are talking about tertiary structure. So hydrogen bonds does involve in secondary structure that will help in stabilizing the tertiary. Disulfide and hydrophobic interaction is for sure, which are involved in tertiary structure. You people might have been confused with B and C and not A and D. The answer is not involved. B and C में confusion हो सकता है because this is involved in secondary structure, this is involved in primary structure. But this shouldn't have been the confusion. Disulfide and hydrophobic interaction. Getting it? Yes? Yeah. Uh, okay, so hemoglobin is an example of a protein with primary structure, quaternary structure, no secondary structure, lipid components. So it's an example of uh, B, which is quaternary structure. Yes. So let's deal with protein folding and oh, sorry. Uh, chaperone proteins assist in what? So chaperones, we did talk about chaperones. It deals with protein degradation or it deals with folding. Is it dealing with DNA replication, carbohydrate metabolism? I think this is easy to answer. Seventh is B. Yeah. Okay. We didn't talk about this though, but if you all know, you can answer. Alzheimer disease is linked to misfolding of which protein? We didn't talk about this, but still, if you know, you all can answer. It is D or A. So Alzheimer deals with amyloid. We also didn't talk about this, but just to answer, Prions disease are caused by. I think we can still say that we did talk about this. The Jacob disease are nothing but Prions disease. The examples which were given in this specific slide. Mm, I couldn't find it. Yeah. So this is Prions disease itself. So if you all can answer, it deals with those examples are of what kind of infection? Was it viral infection? Was it misfolded protein or lipid accumulation? This is easy to answer again. It is C. 9A, 9C, 9B. Oh, all three options A, B, C. Why there is such confusion? It is because of misfolded prion proteins. Okay. So salting out in protein purification relies on what? So we did talk about all other types of stuff. So there is one stuff we didn't talk about. So you can directly answer this as well. So it is we are talking about protein purification and not identification. So UV absorbance will automatically get deleted. So we are having only with left with three options. So this UV absorbance helps in detection and not purification. But these technique can help you in terms of purification. So gel filtration doesn't deal with anything called as salting out. It deals with purifying protein depending upon the size, and antigen antibody depending upon the interaction or affinity. So only one thing which is left in terms of protein purification is solubility difference at high salt concentration. 10th one is A. Then which technique separates proteins based on size? We did talk about it just now, but there was one more method depending upon size. We did talk about this, which is that technique which separates proteins based on size. 11th one, 11th is C, 11th is B, and 11th is C. You all are still confused? C, based on size. C cannot be the answer. Affinity chromatography doesn't deal with size. It just deals with affinity. It just deals with interaction. 11th is B. Yeah. Ravi. Okay. So 11th is B. SDS page is the one which separates on based on size. Uh, so we did talk about this as well. So Bradford assay helps in protein concentration, but it uses what? Does it use fluorescent tags? Does it use radioactive labeling? Do we take absorbance at 280 nanometer, or it has certain dye involved in it for measuring protein concentration? 12th is B. How about others? Yes, it's at 595. You are right. Hmm. So now a bit of proteomics. So what is proteomics? Is a study of what? Is it a single protein function study, or is it an enzyme only enzymatic proteins? Is it entire protein complement of a cell tissue, or is it DNA protein interaction? Yes, the answer is B. Aditi, it's not A though. It is not single protein function. We study each and everything about entire protein complement of a cell. That is proteomics. The 2D gel electrophoresis proteins are separated first by what is the first step in 2D gel electrophoresis? Is it depending upon molecular weight? Is it depending upon the pH, gas electric point, or hydrophobicity, or charge to mass ratio? What is the first step in 2D gel electrophoresis? And the proteins are separated by what uh, method? 14th is A. Is it A? It's not A. You want to check the slide of 2D gel electrophoresis? So in 2D gel electrophoresis, after you take the sample, you will separate proteins by charge in the first dimension and then size in the second dimension. So charge will be involved in what? Now don't answer it as D, okay? 14th, what is the answer for 14th question? Like we are just left with one more. So charge indicating isoelectric point uh, depending upon the pH, depending upon the isoelectric point, the proteins are separated first and then by charge to mass ratio, which is SDS page. So what is the key application of proteomics? Is it gene cloning? Is it biomarker discovery? Is it RNA sequencing? Or is it lipid synthesis? Lipid synthesis can be eliminated right away. RNA sequencing can be eliminated right away. Proteomics will not involve gene cloning as such. So obviously the answer is B. Yeah, you're right. So these are few questions which I had. So I didn't. So I think we can go through more questions in next lecture as well once uh, I can get access with previous year assignment questions also. So that will be maybe we can have revision of both first week one, week two questions together, more questions on that, and I'll also go through the whole week two's explanation, whatever sir has taught in a much more 1.5 hour. Yes, it is mandatory for these sessions also, so that it will be easier for you people to understand. Or if you like, basically, these sessions are majorly for clearing your doubts. If you are having any doubts, so you can go through the sir's lecture, whatever sir is teaching you all, and if you all want to, if you didn't understand any specific part of those lectures, you can come here and ask those doubts, and we are here to try to clear that. So as we are having 2 hours, I don't think like the doubts will go on till 2 hours. So one and a half 1 hour maybe I'll take for just revising each and everything, whatever sir has taught at that specific week, and maybe the other hour or other half an hour we can go through the doubts which you people are having. Yes. Okay. Okay. If you all are having any doubts, maybe we can clear that in the next lecture as we are already past the timing. Still, if you are having any doubts. Okay. Thank you for joining. Where is attendance considered? In the sense, this sessions, yeah, I'll accordingly, like, as this session is recorded, I know who all were present. Yeah. Thank you. Thank you for joining. Bye. Take care.